just to remind you, I will record the presentations. I uploaded yesterday's presentation into YouTube, or no, I'll get my site straight, into YouTube yesterday, so I'll try to do it regularly so I don't get behind. So you'll have this material visually. You can go back if you missed something in your handout or more clarification. You can go to the YouTube video, and I think it's screen capture, so you can kind of scroll through or looks familiar and kind of jump ahead instead of listening to an hour's worth of lecture. We're going to pick up where we left off yesterday, just to make sure that we are familiar with some terminology that we'll utilize throughout the semester. So that's when we apply it, we have an idea. We're going to obviously talk about a cell in much better detail, but we're just trying to get some regions familiar with us at this point. And so we were just talking about how to define something, whether it's inside or outside of the cell. If something is within the cell, which fluid would it be in? Intracellular fluid. We also gave some alternative names for the intracellular fluid. Anybody catch those? Cytoplasm or cytosol. Those terms in this class are okay to interchange with one another. But as for quickly, I'll just may say ICF or intracellular or something like that, implying events or structures within the cell. Conversely, structures and molecules and fluids may be outside of the cell. We call that what? Extracellular fluid. Very good. We learned there really are two big broad categories that we're going to study for extracellular fluid, which include what? Very good. I'll make you proud today. Blood plasma and interstitial fluid. Where is the interstitial fluid located? Between cells. Very good. So it's like on the outside of a cell. So kind of, the word I use and some other people use is bathing. And your cell's clearly not taking a bath, but it's surrounding the outside of the cell. But it's very much different than the blood plasma. Where's the blood plasma? Within the capillaries, all of our blood vessels. You drew it in the capillaries, it would be inside of an artery, or even within the, you can pump through the chambers of the heart or a vein or whatever, but it's in that tubular vascular structure, which is the blood plasma. Now, up till now, you guys probably just called it plasma when you go down to the regional or whatever and you donate your plasma, but I gave you the option to call it your blood plasma. You might know I gave you the caution for you saying blood plasma instead of plasma. There are other kinds of plasma. Very good. Plasma is just a generic term to describe what? Fluid. Yeah. In any fluid, really. In this case, so it is a component of the extracellular fluid. We'll use the term plasma membrane to describe the characters of the cell membrane and the membranes around the vessel. That tells us those membranes are kind of what fluid-like. Okay. So I want to caution you, if you use the word plasma, describe it more, whether it's the membrane or in the blood or whatever the case may be. What questions do you have about differentiating intracellular fluid versus extracellular fluid, or even within the extracellular fluid, determining interstitial fluid versus the plasma? Comfortable with it? So the reason I put this here is because we're trying to link in the organization of the body. We said the person's made up of body systems, which are made up of organs, right? We kept going on down the hierarchy. And what we learned is the organ systems or the body systems, like the nervous system, the reproductive system, name all the systems that we studied yesterday, they somehow contribute some mechanism to make sure that each individual cell's environment, intracellular and extracellular environment, is in that dynamic, steady state. So at some factor, all of our systems are going to make sure individual cells are surviving. So what happens is the local environment controlled by your body system is going to influence an individual cell. If you have trillions, plural, of cells in your body, each one of those needs to be kept alive, and it's by the interactions of the local body system. That kind of re-summarizes what the lady said at the end of the video yesterday, how our multiple systems work together to keep many factors. The term we'll use is homeostasis, separate for all of our cells. And so we've got to see these systems work together to maintain this concept called homeostasis. We used this word yesterday. We defined it, and we said the generic way to describe it is what? Balance. Okay. Factors have to be in balance in order for factors to be okay for a cell to survive. If factors are not in homeostasis, the cell is not going to be in balance, and it's going to be susceptible to disease. It's susceptible to what? If something's sick, it may eventually die. Okay? And then clearly, if a cell dies, what could die? And to get there, if a cell dies, then a tissues, organ systems could fail, right? The whole organism could fail. So we really want to make sure all trillions of our cells themselves are within homeostasis. So the entire organism is within homeostasis. So it's kind of a which came first, the chicken or the egg? Which makes the system survive? The cells or the systems? Cells have to survive so the systems work. The systems work so the individual cells survive. So it's a very intricate kind of dance, if you were, balance to make sure that our cells survive, so our entire body can survive. And it's through this concept of homeostasis. We described this kind of opposite way to define homeostasis. And factors in our body are in a continual dynamic steady state. I tried to elaborate on this yesterday, just illustrating that factors are always changing. That's good that factors are changing, but we need to take them in a narrow range. So the example we gave yesterday was oxygen, right? You take oxygen in with each breath, so that means our oxygen levels are depleting when it goes into our cells. But we replace it, so it's within a safe range, a steady range. We don't ever want to see our oxygen levels plummet to zero and stay at zero. Okay? Because, I mean, that's obvious what's going to happen there in that case, right? So if we look at the definition from your textbook of homeostasis, it says the ability to maintain a relatively constant internal environment. And really, it's the follow-up is despite what happens. Whether it be like stress or whatever. Okay? If you're here in the summer and it's going to get 135 degrees, right? In the sun in the summer, your body is going to work to try to keep your internal body temperature at about what? 98.6, right? Okay? Despite in the Arctic winter that we have, when it's minus 28 degrees, right? Your body's going to try to make sure your temperature is 98.6. So despite what's happening outside of the body, or it's stress, or taking physiology in the summer, or whatever, your body's going to do its level best to make sure all factors, not just temperature, are within a nice, safe, homeostatic range so that your individual cells can survive. And the way it works is there's mechanisms in place to make sure these tolerable ranges are met. This right here, it looks like a bullet point, but this is huge for this semester. Okay, so where we're going today is huge. I'll repeat it in kind of different specific fashions throughout the semester, but the concept is the same. All semester long, we're going to see how different systems work to maintain homeostasis. And there's these different mechanisms or systems that will monitor and control a particular cell or something like that to make sure there aren't those major disturbances. Okay, so an example that you can appreciate is during exercise. If you exercise, you're going to activate your skeletal muscles. Your skeletal muscles, we know, are important for locomotion, but also what happens when you exercise? You're moving. What else? You're getting hot, your heartbeat goes up, and you're sweating, right? Because what we're going to learn in the third unit is your skeletal muscles, only about 40% of the force generated actually goes to mechanical energy. 60% is liberated as heat. Okay, it sounds inefficient. But if you think about, well, if you're cold, what could you do to warm up? Move. Okay, so if you're exercising, your body level temperature is getting really dangerously high. And so we know we need to exercise. We know sometimes we need to run, right? But how does our body deal with that? when we sweat? It cools us off, right? Evaporative cooling, right? Cool, right? Wet at the surface of the body, wind covers and goes across the skin, and it cools off the skin. We also, what we'll learn in class or in lab today or tomorrow, is our blood vessels change and distribute blood differently. When you're exercising, where do you think the blood is shunted to more? The surface, okay, to your skin. So when you exercise, what do you look like? You look red, because your blood is more at the surface than before when you were exercising. Before, it was more internalized to your stomach and etc. And so temperatures are a really great example to understand homeostasis, but what I want to caution you is it's not the only thing that's maintained homeostatically. Everything in your body is maintained homeostatically, besides temperature. So you just list all the things that we listed yesterday that can keep alive. What did we list? Say, oxygen, homeostatically controlled. What else? Nutrients, homeostatically controlled. We could just keep going on, and we'll get some examples in just a minute. But the caution is it's not just temperature that's homeostatically regulated. So some examples you guys just mentioned. Nutrients. This is a snapshot, again, not an all-inclusive list, but just to kind of give you an idea. Our nutrients, like our sugars, our fats, proteins, etc. Our respiratory gases, oxygen, carbon dioxide, nitric oxide, etc. True waste byproducts, urea, ammonia, water, salts like sodium, potassium, calcium, chloride ion, right? All of those have to be maintained within a set point range, homeostatically regulated. So the concentration of some things. And I think on your handout a column or something, so I've got some more things to add on there. So before you turn the page, you can add a few more things. So as well, the pH of different fluids, right? We said you have trillions of cells in your body, and I think if you're a 6 foot tall man, or like me, not 6 foot tall, right? There's a big distribution, right? There's a lot of area to cover. Okay, so what we'll learn is the pH in different regions of the body is not identical. The pH in the blood is not the same as the pH in the stomach, okay? Which is not the same as the pH within the cell, which is not the same as within a mitochondria across the inner membrane. Okay? So all of these factors have to be maintained within homeostatic. So pH is another example of something that's controlled by homeostasis. Temperature we already elaborated on, so overall body temperature, you could tie into your blood temperature, etc. Volume of fluids within a cell, so intracellular fluid, extracellular fluid, so your blood plasma. Going on scenarios as where, there as well. For example, your respiratory volume. So your tidal volume, we'll learn the last unit. It needs to be maintained so that you acquire enough gases and eliminate carbon dioxide at the right rate. Pressure within the lungs, like air pressure, needs to be homeostatically regulated. As well as, your blood pressure should be homeostatically regulated. You understand that if there's a high blood pressure or hypertension in your family, right? You know that if that's not homeostatically controlled, someone's going to be sick and maybe die, right? And so your body is working consistently on maintaining concentration of factors, pH, temperature, volume, pressure, etc. every millisecond of your life, right? From when you're a tiny little one-cell developing embryo, right? All the way up till the final day. So your body is actively just making sure factors are within this set point range. And what we can learn is each system, so kind of tried to start out the presentation with, each system in some way is going to contribute to maintaining homeostasis. So we really kind of outlined this yesterday when we talked about the teleological physiology behind each of these systems. So circulatory system. What do you think, how might it contribute to homeostasis? digestive system, we know clearly has something to do with what? Nutrient absorption and what else? Eliminating waste. What? Eliminating waste and byproducts. So each system at some fashion is going to contribute to overall body homeostasis. Okay? Do I need to go through, do you want to review each of the systems, or do you guys want to just kind of address that each of these contribute? Just review? Go ahead and go on. Everybody's okay to go on? So each system that we listed on yesterday is going to give some type of contribution to maintaining overall body homeostasis, okay? And so we can't just take one system out and expect the entire body to be okay. We can't remove the digestive system and expect us to still absorb nutrients and eliminate waste. We can't remove the respiratory system and think everything's going to be okay, right? They're going to have some important contribution to overall body homeostasis. These terms we'll use throughout the semester, so I'll also put a big fat asterisk to start by here. We'll see it in different fashions throughout the semester, but the theme will be the same. These control systems, we can look at the whole body, or we can look at an entire system, or we can look at a cell, or we can look at the molecular level. All those levels of hierarchy, we can really apply these types of controls to, this homeostatic controls. It does go down to the molecular level. And the control mechanisms can be considered to be either intrinsic or extrinsic. Has anybody used this word intrinsic before? I've heard of it. In what fashion did you use the word intrinsic? Staining on teeth? Okay, so staining in teeth, so there's some, I don't know about this, so there's some teeth that are just naturally, okay. Okay, so she used the word intrinsic in the term of teeth stain, but they're intrinsic, they're really what? Really deep, and so they're within, they're inside the tooth. Okay? The key word to focus on is inside. Anybody else use the word intrinsic? Have you used the word inherent? Use the word inherent? Kind of implying the same thing. It means within. Okay? Within, built within. And so where I'm going is some cells or organs or body systems can intrinsically control themselves to maintain homeostasis. What does that mean? They do it themselves. Some organs can control their factors by themselves. They don't need what? They don't need help. You know of an organ, is a famous organ that even people who aren't even scientists know that is intrinsically controlled. We just didn't use the word. What's probably the most famously known organ in your body that can intrinsically regulate itself? The heart. It has a what? What does the heart have to allow for it to control its actions? What do we call it? They're called nodes. That's right. The layperson would call it someone on the street, or if you go into 7-Eleven, we call it a what? Pacemaker, right? So the heart can set its own pace without the nervous system, without the endocrine system, whatever. So the heart is an example. You know, I should go ahead and add that. I think I've got some images that the handout. So the heart is a really famous example of an organ that's intrinsically regulated. It can regulate its own pace. What kind of pacemaker size? The chart really called nodes. The example I have here in the presentation is the detection of oxygen levels in muscles. If oxygen levels deplete in a skeletal muscle, there's going to be more abbreviation to blood flow. If there's more blood flow, there's going to be hopefully more oxygen. So the muscles and cells can change the diameter of the blood vessels in the muscle to make sure that there's more blood flow in there. It intrinsically just causes the muscles to dilate and the blood vessels to bring more blood. So our skeletal muscles can do this. Our heart can do this. Other organs that you can add in the list that we're going to see this semester, besides the heart, include what's called your visceral smooth muscle. And so you can say some smooth muscle in your stomach is intrinsically regulated. What we're adding there is the heart. We're adding in there some smooth muscle, not all, is intrinsically regulated. So for example, when you eat, what happens to your stomach? First it stretches, and that stretching causes it to begin to turn. So just food's in there, stomach doesn't go, right? And by itself begins that. So the heart can do this. Some smooth muscles can intrinsically regulate themselves. Another organ that we'll study at the very end of the semester are your kidneys. The kidneys can also intrinsically regulate blood flow through the kidneys. If there's an infection in the body, we're not in homeostasis anymore. If it opens up a new can, if you will. And so, depends on where the infection is, the type of infection. That's a very difficult question to simply answer, basically. But the way to simply answer it, if there's an infection, you're not in homeostasis, this isn't prevailing. And so, intrinsically, it's not working. Very good. What other questions do you have about how to define intrinsic or some examples of intrinsic homeostatic regulation? If something can be intrinsically regulated, it probably can be extrinsically regulated as well. Extrinsic regulation means what? Something outside of that organ or cell or tissue or whatever is doing what? Regulating it, controlling it, telling it what to do. Yes. It's depending on something else, some other organ or molecule, tissue, whatever, to control it. To make sure it's in homeostasis. Before I move on, to better clarify this, I want you to be aware that just because something's intrinsically regulated doesn't mean that's all that it's got. If something is intrinsically regulated, it can also be extrinsically regulated. Let's go to our scenarios. The heart. The heart can control itself, but if we get up and we go do our exercise, we go run outside 3 times around the courtyard, the heart can't handle that by itself. What does it rely upon? If I begin to just start running, what's regulating the running? Muscles, cardiovascular system are involved, but what tells your muscles? The nervous system, the brain. So your nervous system can control the heart extrinsically. Hormones can control the heart extrinsically. The pharmaceutical drugs that you take for your blood pressure can control your heart extrinsically. Yes.
So you have an intrinsic pacemaker in the heart.
Yes.
And then if it stops beating, and then the cardiologist puts the pacemaker back.
Yes. 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 The question is, well, let's say, and also we can also say, well, it goes into the disease state and doesn't fall into normal. But I can answer that a little bit better because we're putting things back to normal. The question was, well, let's say we've got our built-in pacemaker cell. It's called the SA node. Let's say you've got disease and it stops working. You go to the doctor and they do install a pacemaker. That pacemaker is now extrinsically regulated. regulating. It's mimicking what would normally be inherent or intrinsic, but it's not the cells of the heart, it's extrinsic. And that's in a disease state, not desired to have an extrinsic pacemaker, but it's good to know that it's obvious. So if something's intrinsically regulated, is that all that it has? No. It's got to back up and override by what type of controls? Extrinsic controls. So the question is, well then what the heck are the extrinsic controls? We already listed one, which is the nervous system's a huge, important player for extrinsic regulation, as well as the endocrine system. The nervous system and the endocrine system are our major extrinsic regulators. This is also huge for the rest of the semester. I want you to remember this all the way to August through the semester. And instead of applying it, I'm giving you examples that hopefully make sense. The nervous system will control extrinsically organs, and if we try to quantitate it in a fast, right, fast, really fast, within milliseconds, right, snapping of a finger fast, right, control. The nervous system can extrinsically control something very quickly, very precisely. The example is like if I exercise. If I start doing jumping jacks, which I won't, I'll save you, right? If I were to start doing jumping jacks over here, nervous system can actually control those muscles quickly, like be able to jump right away. Conversely, the endocrine system probably controls things how? Duration-wise. That take a little bit longer. And a little bit longer in your body is like more than a few milliseconds, so a few seconds, minutes, hours, days, weeks, whatever. And so it's not as long necessarily, like, it doesn't have to be days. It could be longer than a few seconds. And the endocrine system may kind of take in and prolong that activity. That may last hours or something like that. But it may not take hours to kick in with you. Does that make some sense? So the nervous system will control extrinsically organs and cells, quick, precise, rapidly. Endocrine system can also control the same organs, but if we need to control it for more than like a millisecond or so, the endocrine system will take over to control something. So like the heart, right? I can control the heart. Hormonal is not extrinsic. Kidneys can be under hormonal control. They can be extrinsically controlled by the nervous system, by the endocrine system. My digestive system can be controlled by the nervous system. My digestive system can be impacted upon by the endocrine system. But if something's not intrinsically controlled, it relies upon what? Extrinsic sources. The nervous system tends to be more what? It's certainly an extrinsic form of control, but it tends to be, speed-wise, rapid, quick, fast. Okay, faster than I can snap my fingers. The nervous system, right, can control something. Endocrine system, what? Takes longer. Okay, so a few seconds, minutes, hours, whatever. You're pregnant, months. The endocrine system's controlling that. And then after birth. These are the two I like to highlight, but really after perspective from last semester, I want to go ahead and put this here, a third important extrinsic regulator, because we'll address it in the endocrine system. I was like, why don't I just put it here, too? A third important extrinsic regulator is called humoral, H-U-M-O-R-A-L, humoral. Is a third type of extrinsic regulator, but I won't really talk about it much, and so that's why I didn't put it up at the beginning of this semester. Hindsight, I think I need to add it. Do you know what the heck that is? It can be an example, because where is the immune system? Well, it is, and in high proportion, it's in your what? And in your blood. and then more specifically in the factors in the blood. So factors in the blood can humorally, extrinsically regulate other organs. And humoral is the third thing that we're adding. And you notice the word factor is just about as generic as you can get, right? So in other words, it's not a hormone, and it's not a neurotransmitter. It's not something from a gland, not something from a different gland, and not something from a nerve cell. So for example, like your iron. Calcium levels can influence hormone release from your parathyroid gland. So the gland itself is controlled by a factor in the blood. It's humorally regulated. The secretion of your kidneys is humorally regulated by blood levels of sodium. Sodium's not a hormone, and it's not a neurotransmitter. It's in the blood. Does that make sense on the third factor? So some organs can also be controlled extrinsically by just what's in the blood, or some specific thing in the blood. That too would be obviously a lot longer than taking nervous. It'll take a little bit longer. So words we're getting familiar with the next 8 weeks. Intrinsic means the organ is controlled internally by itself. Or it may be also, it's hard to say, extrinsically regulated. So some factor what? Outside of it, like say the nervous system, which provides what type of control? Quick, rapid. Or maybe the endocrine system that will take over a long-term, more long-term. It's kind of a generic way to describe it. Or even what? Humorally, which means by what? Factors of life. Any questions before I move on? We gave some examples of intrinsic control. The pacemaker, we'll come to this in the third or fourth minute. If you look at the heart, there's a set of cells that regularly generate what we call action potentials that cause the heart to beat. So we gave examples of the heart. We gave examples of the smooth muscle, kidneys, how they're all intrinsically regulated. Extrinsic, am I okay to move on? Extrinsic controls, for example, hormones regulating your nutrient levels, like your blood sugar, you're familiar with, your blood glucose. You probably know like insulin extrinsically regulates glucose. Anybody know the antagonistic hormone to insulin to elevate your blood sugar levels? Glucagon, very good. So don't sweat it yet, we'll learn it in the third year. We'll look at something on the sweat it then. So your blood sugar levels, they don't magically monitor themselves, hormones have to control. This too is the common theme that we're going to see besides intrinsic, extrinsic. We're going to see how it works and how something is monitored to be maintained homeostatically. There's got to be sensors or receptors. Right? I think I have you writing down the word sensor. Another term that I'll use this semester is receptor. You can put sensor/receptor. This will be really repetitive for this semester. In order for a particular factor to be maintained homeostatically, whether it's intrinsic or extrinsic, whatever's monitoring has to have the ability to detect what it's monitoring. Does that make sense? And the way it works is by utilizing sensors or receptors. Whether they be entire cells or proteins in a membrane or whatever, it's designed to receive a particular mode of stimulus. Whether it be like glucose in the blood or temperature in the blood could be the stretching in the stomach or whatever the case may be. There has to be a way to physically detect that motor stimulus. And the way it works is called a sensor or a what? Receptor. Could be a molecule. It could be a what? The sensor itself could be a molecule or like a protein in a cell or what? Just y'all understand. So it could be just a protein within a cell, acting as a receptor. It could be an entire cell that works to determine physical stretching or something like that. It could be a molecule or an entire cell, maybe a tissue or something, monitoring your factor. So the sensor, the receptor, all it does is, what is its job? Monitors. That's a good way to describe it. The receptor monitors. That's all it does. All it does. It monitors. It monitors. What does a monitor need to do? Were you a hall monitor in grade school? Well, you have to, they don't have as much order though, so what do? Watch and tell. Okay? So the receptors, that's all they do. Is they watch and report. Chemically, they hit on hormones. So they watch and report. Who do they report to? The sensor's got to report to something that has something called a control sensor, or they call it an integration sensor. Here in the handout, I have an integration sensor. The control center, well, kind of applies a lot, except for what? We got some control, power, and authority. But it makes its decisions based upon what? The incoming information from the sensors and the receptors. So the control center, the integration center, takes the sensor information, looks at it, and compares it. It says compares with other information. That's just how it's laid out on your handout. Let's say compares, integrates information. Let's use the word instead of other info. Let's use the word set point. Let's scratch this word and put the set point. We're just going to put in the word set point. The control center compares the incoming information. The set point. What do you think that word set point? Here's the information. Your set point. What do you think set point is? Where it's supposed to be in our balance. If some factor's not in set point, what do you think has to happen? Changes have to be made. How do we bring about changes? It could be intrinsically or it could be extrinsically. So let's get third player. What's the third player? The receptor are actually it's called the effector organ. It could be an organ or a cell or a molecule. There's going to be an effector. It could be a cell, it could be a molecule, it could be did it know to do any work? The control center. The control center tells it what to do. So the integration, or the control center, monitors information based on coming in from the what? Sensors and receptors. Compares it to set point. If that factor that receptor is designed to monitor is not a set point, the control center will activate what? An effector. An effector should hopefully use action to bring that factor hopefully back to set point. We'll know if it's set point because we're still continuing to send some monitoring. We're continuing to report. We're continuing to adjust. So what is the role of the receptors or the sensors? Monitoring and reporting to the control center. That's all they do. Okay, here's the data. There you go, control center. The cells or the molecules that act as the control center then take this value and see if it's good or not. See if it's a set point. If it's not, it will activate an effector, whether it be a cell or molecule, whatever the pathway is. And the effector will bring about the changes, hopefully to bring the factor back to the set point. We'll know if it is because its sensors are still sensing the molecule. I'll just turn on. So, the question is, what was that? Right? Hopefully not too much. And the question is, how does it work? Well, there's two primary ways at which this works. And the more familiar, kind of easier one to understand is this concept called negative feedback. Negative feedback, that's a negative connotation, right? Negative. But I want you to equate the word opposites. When you see the word negative feedback, I want you to think about opposites. So in other words, if a receptor monitors something and it's below set point, negative feedback will kick in and bring that factor back up to set point. So negative feedback, there's an opposition. To bring the factor back to set point, back to maintain what? Homeostasis. So classic example that we gave, temperature regulation, right? If I got too hot, negative feedback should kick in here and allow me to cool down. If I'm hungry, my blood sugar level, right? I didn't eat breakfast even though I had like 3 hours to eat breakfast this morning, right? My hormonal pathway says your blood sugars are low. I should have a hormonal pathway to release this hormone. Glucagon, bring my blood sugars back up to set point. So negative feedback is fairly easy to understand. If you're out of set point, bring it back, whether it be up or down. And bring it back into set point. Some examples of negative feedback. I'm going to come back to this image in the body. For example, just putting an application in your body temperature. If I become too hot because I'm exercising, I have specialized nerves that have specialized temperature receptors on them that will communicate with a set of cells in my brain, in the region called the hypothalamus, that say, well, this temperature is too hot. That's above the 98.6 that I'm okay with. And it'll go out and control some effector organs. You guys already identified the effector organs in this pathway, your sweat glands and your blood vessels. Their actions will allow for the body temperature to do what? If I'm too hot, hypothalamus, yeah, the effect is to cool it down. So the blood vessels will dilate at the skin. I'll sweat. You said evaporative cooling. So those are the examples of the effectors. But it's not the skin. It's not the skin itself knowing to control your blood temperature. It's coming from the brain. The brain knows what's good for you. And it has a what established? The set point established. But the brain's not out on your skin. It's got to have what out in your skin to monitor? Receptors, specialized temperature receptors in the body. Does that make some sense? If I illustrate, if I'm too hot, I'll cool down. If I'm too you're in now a disease state, and then you're susceptible to die, right? So if you go hop in a fire, your body will try to pull you down. Fire's hotter than your ability to maintain. So yes, at some point, there's
But
always
it'll
some.
always try one more time.
Not today, yeah. No, it doesn't take a day off. It doesn't take a day off, and it's going to do its level. Your body's going to do your level best. Heat factors and homeostasis, despite what you do to it. And think about what we do to our bodies. Okay, we wouldn't hopefully hop in a fire, but we do take drugs, and we do drink alcohol, and we do reckless things to our bodies, but we're still live, right? And function, right? The body's going to do its level best to do what it's designed to do, to keep you in homeostasis. But if it becomes too much, too many bad drugs, right? Etc. Those are extreme examples, but hopefully to illustrate. So negative feedback does what? What's the key word? Opposes something to bring it what? Back to set point. So I have a simple image to illustrate of the brain's controlling your body temperature. An example that we'll use, I think your book goes into the pathway today, but we won't cover this until the 4th unit. We'll talk about blood pressure regulation, but your blood pressure is regulated by negative feedback. If your blood pressure elevates, hopefully it brings, your body will make a mechanism, bring it back down to set point. The even just between lying and standing down will cause a huge fluctuation in your blood pressure, which we don't want, so we need to make sure it stays consistent, about that 120/80, whatever your blood pressure is. Another example, the antagonistic relationship between insulin and glucagon. To bring your blood sugar back to set point. If it's too low, glucagon brings it up. If it's too high, insulin brings it down. Again, this pathway we'll cover in the 3rd unit, so you don't need to know that pathway. I'm just trying to illustrate some things that would be familiar to you. Are you okay on negative feedback? Question? I gotta know, I don't get it. What did you miss? The effector. The effector is the structure or cells or whatever that physically does the work to bring the factor back to set point. So for example, if I'm hot, if I exercise, I'm getting hot, I'll have receptors in my skin, my muscle will start saying, you're going to temperature to the brain. The receptors will communicate with the control center and they'll have to tell them, it's time with allergy. That's too bad. That's too high. It'll communicate out to the effector organ. In that case, the effector organs would be blood vessels. They would change their status, and as well as your sweat glands. They would become activated, and they would sweat. And so in that case, the effector organs, the sweat glands, and the blood vessels, they're the ones physically doing some action, and the effector is in the body. In that case, in the hormonal pathway, the effector, if we're trying to maintain blood sugar levels, the effector would be the hormone insulin or glucagon. So it's a thing causing an action to bring the factor back to set point. The effector only does its work when what tells it to control it. You okay with that? Anybody else have any questions for us? So in negative feedback, we oppose. If there's negative feedback, there's probably positive feedback. There's one that sits with that in physiology. So negative feedback opposes, positive feedback must do what? Goes in the same direction. Actually continues something. Positive feedback is not as prominent, but it's useful, where we continue something. We can appreciate positive feedback in extreme scenarios. So, for example, everybody loves this. It's a very visual example of positive feedback. Is during childbirth. Just, anybody having kids? Everybody had given birth to a child? Yes. All right, so yeah, like 50% of the class. Okay. So, in that scenario, or if you're maybe your father, you've been there and it's going down, right? Do you want that thing to be controlled under negative feedback? Oh, let's start it. Oh, no, stop it. No, start it. Go, stop it, go. Is that good? until we reach the desired result. Okay, so during childbirth, that's under positive feedback. We continue it. Oh, let's keep it going. Oh, let's keep it going until the baby's born. Let's keep it going until what? What else has to happen? Well, since it's delivered. And then let's keep it going until what? Until that uterus shrinks and all that massive bleeding, ridiculousness is kind of shut down. Okay? And so positive feedback controls that. Eventually, we don't need to continue contracting in the uterus, right? Eventually we'll turn them off. But the whole point is to deliver the baby, to deliver the placenta, to start healing. That's a great example. It's under the influence of this hormone called oxytocin. And if you're in the hospital, you may have been administered something called pitocin, which is synthetic of that. And it promotes uterine contraction, so it continues it. In the female, it promotes uterine contraction. We'll look at its hormonal pathways in the third unit with the imaging. But, that's not the only scenario. Examples is the clotting mechanism. Think about that. If you start to form a clot, don't you want to continue it until the clot is fully formed? You don't want to just kind of start the clot and then still bleed out, right? Continue the clot. Prior to a woman's ovulation, huge luteinizing hormone or LH surge, just spikes, and we'll talk about it in the endocrine system. Follicle will rupture, egg will be released. So it's just, right? But it's huge spike, positive feedback of luteinizing hormone prior to ovulation. Male ejaculation, another example, positive feedback mechanism. If you've ever vomited, all those muscle contractions, how they continue, those are examples of positive feedback mechanisms. So, in summary, positive feedback will do what? Enhance or continue something. Negative feedback will oppose something. I want you to look at these. I know it's across both pages, but I want you to look at these. I mean, these are not technical. These are something I've tried to draw on the computer, so it's not great. But if you can look at some scenarios, say this is some factor at set point. Let's say this is LH levels in a female, but she's about to ovulate. And so the LH levels will increase and then increase and increase and increase. And so we clearly can see what? A rise. Okay, so we're getting further away from set point. We're continuing something. Does that make sense? Let's go back and look at the image that I quickly went over for negative feedback. Here we have the same scenario of some set point. Okay, let's say this is blood glucose levels. Okay, we've got our range. We can clearly see negative feedback, right? Where it's high, I want to bring it down. When it's down, I want to bring it up. But hopefully it's in a dynamic, steady state. I don't see crazy fluctuations, pretty consistent. Let's scrutinize it. We'll look really close. One, can you see the opposites? Opposites. How do I get from point A to point B? How do I get to set point? A vector is going to make a change. But look really closely. I'm continuing. I continue to decrease. What really drives negative feedback? Positive feedback. If that's not crazy enough, right? If I'm too high, I need to get low, I need to continue to drop. And I need to continue to drop. And I need to continue to drop, right?
So couldn't that be the LH one you're talking about, like right there, like because that must happen when she's been ovulating or something?
At the end of it, yes, but not during the decline of it. So, for example, let's just go with something that you're okay with, like blood glucose sugars levels. So if my blood sugar levels are too high, I need to release. If it's too high, I need to release insulin because I want to bring my blood sugars down. But I don't want to release just a little bit of insulin because it would just drop a little bit, right? I need to release a little more insulin, drop the glucose, release a little more. So if you're really scrutinizing it, to drive negative feedback, you need positive feedback. Something until you reach that bottom range. more time. Negative feedback does what to a factor? Opposes it, to bring it back to set point. Positive feedback will continue something and enhance it until we're actually deviating away from set point for a period of time. And it'll turn off by negative feedback once the goal is attained. I think is where you're going. You're not going to indefinitely, once you ovulate one time, you're not going to for the rest of your life release elements of crazy positive feedback. At some point, normalcy will stop vomiting. Baby will be delivered, etc. And then negative feedback will fight with that. Are you okay with the difference between negative and positive feedback? Any questions? Intrinsic? Inside, extrinsic, outside. Our major extrinsic regulators are the nervous system, endocrine system, and we added in there a few moral factors. Very good. Let's go ahead and jump into chapter 2. We have about 15 minutes so we can introduce it. Chapter 2 is a review of cell structure and cell structure function. Okay, so we've got to review. We're not going to be able to recognize, just pick these things out, but we've got to know their function. So we're going to talk about a generic cell. We're going to keep these cute little circle, triangular, rectangular, whatever cells. We're not going to say nervous tissue, muscle tissue, whatever. That's what we'll get into when we get into their respective systems. Or we're going to talk about whether it be a nerve or a muscle or blood cell or whatever. You're going to find these common features about all of our cells. Okay? In the chapter, you'll notice the opening text gives you all the basic biomolecules and cell division, all that stuff. I'm not going to hold you accountable for it. I'm presuming that you're familiar with those things. We're just going to review the primary structures. So we're going to need to know their function. So protein synthesis, whatever, you need to, that's on your own. You don't need to write this down. Those are omitted from the reading pages. So the reading pages you'll notice really include this. You don't remember the different types of macromolecules or how proteins are made or cell division, but you're ready to go forward in the class to kind of review that. I always like to take a short trip down history lane to talk about cell biology in general. Not a really all-inclusive list, but just a kind of major peaks in the history of understanding cells. And if we go back to the 1600s, you think about this, this is a long time ago, a few years ago, centuries ago, and people were very much curious about life and biology and origins of life back then as we are now. And Robert Hooke, you've probably heard of him before. You heard of him? He utilized a microscope, and with his magnifications, he was able to thinly slice corpse tissue, look at it under the microscope, and he was able to observe these little rooms. He called them rooms, or cellulars. Just like we call it prison cell, right? It's a little room, and not necessarily in the biomolecule kind of living fashion. He just saw these repeated structures. And remember, he sees the structure, he doesn't know the function. He just sees these little repeated structures. And so he's looking at plant tissue, and he coins the term cellule, or what we use, cell. We move forward in time. You got to keep, as we advance in time, what else are we advancing? Microscope technology. And so the microscopes are improving in optics significantly. And even to this day, our microscopes continue to improve. And so we move forward a few years. Leeuwenhoek looked at actual living cells. And so his optics were good enough that he was able to take pond water and look under the microscope and see this little amoeba and other creatures moving about. So he saw that there are living things besides plants and animals on there. He was able to look at them. They're microscopic with his optics. We move forward to the 1800s, early 1800s, Robert Brown, his optics improved enough that he was able to look at not only saw just the cells, but he saw a compartment within the cells. And he called it the kernel, or the nucleus translates to the nucleus translates to kernel. So within each cell, he saw a kernel. He didn't know the what. He had no idea the magic that happens inside of that little kernel. Didn't know about DNA. Didn't really know of. People are starting to stumble upon genetics or heredity. No direct connection. Nobody said there's a cell, and inside of it there's a kernel. We now call it the nucleus. We move forward again in the 1800s. Schleiden and Schwann were also studying tissue. Schleiden was working with plant tissue. Schwann was working with animal tissue, which was a big leap. Up till now, most work was with plant tissue because plants are easy to obtain. Now, it's not really unethical to go kill a plant versus a person or something like that. And plants have those cell walls. They're easy. If you have your bio class, you look at the plant tissue and you're like, cell, 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 right? You look at the animal tissue and you're thinking, what is this, right? And so plant tissue is a lot easier to look at under a microscope when compared to animal tissue. But Schwann was beginning to look at some animal tissue. And between their work, Schleiden and Schwann kind of put their neck out there, if you will, in the scientific world. And they concluded that, Schleiden concluded that plant tissues are made up of individual cells. The entire big structure that you see is made up of repeated cells. He also went on to say, well, I think that big multicellular structure actually originated as a single cell. I think that cell divided and made this big thing. And we would think, okay, yeah. No, I don't usually hold a history. I think it's interesting. I hope you find it interesting as well. And in general, those kind of questions are really easy. It'll be all be accountable. Okay. I won't point it out if I don't think it's interesting. Okay. I'll try not to. So plant tissues, all right, are made of cells. They do come from a single cell. And then Schwann went through and said, well, I think also animals are made up of multiple cells. And I also think that animals arose from a single cell. So we're beginning to see some synonymy between cells of plants and animals. With their work, they were able to comprise this thing called the cell theory, which we addressed on, I think, yesterday. We mentioned it. Okay? And so 1839, the collective work between Schleiden and Schwann, what they said is all organisms. They put it in historical context, the word all up till now is plant animal. They didn't understand bacteria or any other kind of organism at this point. So up till now, they just said all organisms. If something is considered to be living, it has to have what? A cell. Furthermore, the cell is the basic repeating unit for all living organisms, whether it be plant or animal, and we now know bacteria, archaea, etc. The two tenets sound very similar, unless you put it into practical, like over-exaggerate something silly. The pen in your hand, does it not have your cells on you? Your cells on it? It does. It has one or more cells. So is the pencil living? No, because tenet 2, what? The pencil's not living because why? The structure is not repeated cells. So these are the first two tenets that were proposed. The cell theory was then modified later by Virchow, and they went ahead and said, yes, all cells do come from pre-existing cells. We say life begets life, basically. And yes, this will be on the test. This is very important foundation for this course. This is the theory which I mentioned yesterday that we're going to base this course on, is the cell theory. We're going to say something's living, it's comprised of a what? A cell. This is a thing that I said is debatable. You may not accept the cell theory, and that's perfectly fine. But if we don't accept it, it kind of unravels all of this concept that we're going to study this whole semester. So it really is a good, solid scientific theory. What is the opposing theory to the cell theory? About what life is. What is living? Well, spontaneous generation, I don't think would necessarily, well, it just follows this third tenet. What else opposes it, besides, in addition to the spontaneous? I don't know if anybody still supports that. Or maybe, there's always somebody. Do what? Did I hear it? Some people believe that viruses are living. But viruses aren't what? Well, viruses aren't. Well, someone could, you could go toe to toe with someone that yes they are, no they aren't, yes they aren't. What would you, if you believe in the cell theory, what would you say? Viruses are not. They're not made of cells. They're not. Viruses aren't cells. So they don't fall into the cell theory. So if we're going to take a steadfast view here, we say viruses are not living. But they clearly have an impact upon life. Depend upon them and are affected by them, etc. So we're going to go with the cell theory because it's going to support how our tissues work. We'll run with it. What questions do you have about the cell theory at this point? All right, so if we look at a given cell, the cell is described as being dynamic. Well, we already used that word before, so it's always what? Changing, right? When you look at the book and you see your cells, and they're cute, and even the handout that I can see here, right? Handout number 1. It's cute, it's big, it's colorful, it's like right. Our cells are not cute and pretty necessarily. They're not colorful. They're not static. They are busy, working, living, single, compartmentalized structures, right? They're always changing factors. Mentioned as well already this morning that there are trillions of cells within your body, right? Like between those 4 primary tissues that we identified, from the muscles and your nerves and your connective tissue, etc. We've got lots of different cells in our body. Within those primary tissue types, there's collectively about 200 total variations of cells that you might find in your body. Although some primary functions of them are the same, there's going to be clearly shape and size and thusly functional differences between all of our cells. So for example, a red blood cell looks completely different from a nerve cell, which looks completely different from a fat cell, which looks completely different from a muscle cell. But they're all going to have some similar features. They're going to have a membrane, a nucleus. Most of them have a nucleus, DNA at some point, and a mitochondria. There's going to be some similarities, but some slight modifications in structure, and therefore some modifications in functions. I got to throw it out there. I'm going to say it all day because what? Structure dictates function.
Is that what we talked about, or not talked about, the red in chapter 1, like the stratified?
Did it go into the different subtypes? So yeah, the stratified, the squamous, epithelial, whatever. Yeah, those are all different subtypes. And so I could talk about just my skin, or if you get really microscopic and say, so my tubular, are stratified, tubular, whatever. And the case may be. Very good. Sickle cell is a great example of structure dictates function. Our red blood cells are normally how? And disc shape. In the disease state, in the sickle state, it turns into a sickle, a hook. We change the structure, and now what happens? Can it flow through the blood very well? It's hung up, tissues become painful and damaged. Very good. All right, so just some examples of, here is a good example of it. Here's a normal shape of a red blood cell in a biconcave, and we'll cover that in chapter 15. In the first panel, the second panel, kind of a pretty image of a nerve cell. You can see its processes, very unique. And then the last panel, a sperm cell. Okay, so all these cells have membranes and intracellular fluid and proteins and etc. But they're built differently, so they certainly, what? They're going to function differently. We're going to conclude our discussion today. However, on tomorrow, we will try to get through most of the cell structures as best we can. I know your handout's not due until Tuesday, but I would suggest you complete at least the first page of handout 1 tonight, so that lecture tomorrow will go really slow.